Hello, I am Mikey Katz, and my group members are Rodney Wright, Victoria Echeverria, and Marissa Quintana, and this is our cell division restaurant project. Cells divide for multiple reasons. Some of the reasons could be to replace damaged cells, the organism could be growing, the cell is becoming too large, and for reproduction.
Do most of their growing during the G1 phase. In this phase, cells increase in size and synthesize new proteins and organelles. The G in G1 and G2 stands for gap, but the G1 and G2 phase are actually periods of intense growth and activity.
S phase is the phase of the cell cycle in which DNA is replicated, occurring between G1 phase and G2 phase. Since accurate duplication of the cell is important for cell division, the processes that occur during S phase are tightly regulated and widely conserved.
The G2 phase is the time during which the cell replication its organelles for preparation for mitosis. Not only does the DNA need to be divided, but so do the organelles. G2 is the last chance for the cells to make more protein preparation for division. The cell has twice the amount of the DNA during the G2 than it did during G1.
The first phase of mitosis, prophase, is usually the longest and may take up to half the total time required to complete mitosis. During prophase, the duplicated chromosomes condense and become visible. Centrioles move to opposite sides of the nucleus and help organize a spindle. Spindle forms and DNA strands attach at a point called their centromere. The nucleus disappears and the nuclear envelope breaks down.
Metaphase is the third phase of mitosis. During metaphase, the cell chromosomes align themselves in the middle of the cell through a type of cellular tug-of-war. The chromosomes, which have been replicated and remain joined to central point and called the centromere, are called the sister chromatids.
The third phase of mitosis, anaphase, begins when the sister chromatin separates and begin to move apart. Once anaphase begins, each sister chromatin is now considered an individual chromosome. During anaphase, the chromosomes separate and move along spindle fibers to the opposite ends of the cell. Anaphase comes to an end when this movement stops and the chromosomes are completely separate into two separate groups.
During telophase, the chromosomes, which were distant and condensed, begin to spread out into a tangent of chromatin. A nuclear envelope reforms around each cluster of chromosomes. The spindle begins to break apart, and a nucleolus becomes visible in each dotted nucleus. Mitosis is complete. However, the process of cell division still has one more step.
During cytokinesis in most animal cells, the cell membrane is drawn inward until the cytoplasm is pinched into nearly two equal parts. Each part contains its own nucleus and cytoplasmic organelles.
Cytokinesis in plant cells proceeds differently. The cell membrane is not flexible enough to draw inwards because of the ridged cell wall that surrounds it.
Instead of the structure known as the cell plate forms halfway between the divided nuclei, the cell plate gradually develops in the cell membrane that separates two daughter cells. A cell wall then forms in between the two membranes, completing the process.
A cell cycle is highly important in organisms, as if it were not present or if it were to stop suddenly, life would completely end. Cell cycle is the replication and reproduction of cells. If an animal were to have a severe injury, it would need the cell cycle to replicate cells to replace those that were damaged, and the injury, and using the cell cycle, it would heal themselves.